Everyone, and welcome to our session today on fellow Newnhamite, June Lindsey, and the contribution that she made to the Double Helix project. My name is Margaret Campbell. I read French and German here at Newnham, and I'm delighted to be back in college on this beautiful Sunday morning and on this weekend of a really splendid festival of the arts and sciences. I actually live in New York now, and I'm also chair of the US Committee for Newnham Alumni in the USA. It's my pleasure to introduce our speaker this morning, Alex MacKenzie, who's going to talk about the life and career of his friend, June Lindsey, and on the Zoom, we also have her daughter, Jane Lindsey, listening in this morning. June was yet another of the remarkable Newnham alumni and scientists that we have had. And she passed away at the age of 99 in November 2021. She was another of those pioneers in the discovery of DNA and the double helix, but one whose very significant and crucial contribution has been overlooked. Hence the reference in our title to the other Newnham alumna. Alex MacKenzie is a pediatrician and clinical scientist at the Children's Hospital of Eastern Ontario and CEO of the Research Institute, and he's going to talk to us about how he got to know June Lindsey and about her life and career. He's going to talk for about 30 minutes, and then we'll have time for Q&A when we'll pass a microphone around the room for any questions you may have. Alex, over to you. Alex, I think you're muted.
Hi there, everybody. Thanks so much for that gracious introduction, Margaret. Hi, everybody. Alex MacKenzie here to tell you about my friend, June Lindsey. And a bit sciencey in places, so I apologize in advance. But here's a picture of June with her husband, George, taken in 1951 or thereabouts when they were living in Ottawa, her son Robin tells me that they're about to move to Montreal at that stage. And this was actually at the tail end of her career, although she was a young mother and housewife in Ottawa. She had been working at the National Research Council. At any rate, they were friends of my in-laws, George and Eve Hampson, and I got to know them peripherally since the 1980s. George was himself a Cambridge physics grad, a brilliant man who was the descendant of William Lyon Mackenzie King, one of the prime ministers, and was the architect of the defense policy in Canada in the 60s and 70s, and was largely responsible for divesting of nuclear weapons, which is not a bad thing. And so he really was, he also was incredibly numerate and more or less invented the area called sabermetrics, where you look at baseball with through a statistical lens. It was a subject of a movie called Moneyball some years ago. So he was an Order of Canada. And so he was really the one that I was most aware of. And June was a lovely, soft-spoken, but it has to be said, and it's probably a reprehensible term these days, a wife of, as much as anything. Time went on. George passed away in 2011, and then fast forward to 2017 to my wife Beatrice's mom Eve's 90th birthday, and June was there, still living alone. She was in her house, but at 95, still driving, still very much with her considerable faculties. And I asked her in that faintly patronizing way that one asks the elderly, which as the years go by, I'm revisiting that approach. We should be more polite to the elderly at any rate, because people are asking me that way these days. At any rate, what she did And she said she did crystallography at Cambridge. And I think the matter was left there that evening, but it did pique my interest because Cambridge, and in particular the Cavendish laboratories under Sir Lawrence Bragg, was Mecca. It was more or less where this idea of crystallography was developed and refined. And it's the arcane art for the non-scientific in the audience of passing X-ray beams through a crystallized structure and looking at the exotic shadow pattern called the diffraction images, and using mathematics, which is far beyond me, you can discern the actual structure of the molecule through which the X-ray beams are going. There's a story that Bragg was walking along the banks of the River Cam one evening and watching the sun slant through the serried ranks of trees along the bankside when he had the epiphany that this could be used to deconvolute the structure of complex molecules. I'm not sure if it's true, but it's a lovely tale. But anyway, we see here actually the diffraction pattern taken by another Newnhamite, Rosalind Franklin, the famous photograph 51. And it's precisely these sorts of images that June was looking at. And so either that evening or the next day, I did as we all do these days, Googled her, or more to the point, did a literature search. And much to my amazement, I found this single author paper in 1948 with June, Broomhead as she was then, elucidating the structure of a molecule known as adenine. And then another paper just 3 years later, her single author once again delineating the structure of a molecule known as guanine. And I was thunderstruck, to be honest. This quiet, soft-spoken woman who I'd been so inquired patronizingly of what she did, had done this rather remarkable work. And just to pull back the lens a bit, here is the complement of DNA in a human cell. It's 3 meters of DNA, which is, sorry, I'm on call for the hospital. I have to take my thing on mute here. I apologize. 3 meters of DNA coiled into every cell of the body. And if one looks more closely, it is the famous double helix we can see that it's made of. And even more closely still, there it is, basically deoxyribonucleic acid. The deoxyribose are the ribose sugars without the oxygen, which form the 2 backbones on the outside, alternating with phosphate molecules. And then the inner workings where all the information resides are these 4 compounds known as nucleic bases, the so-called purines and pyrimidines. And June had single-handedly elucidated the structure, the physical structure of the 2 largest, most complex ones, adenine and guanine. These are the actual units that Watson and Crick were using in order, when they were trying to put the jigsaw puzzle together. And she was responsible for actually generating those physical structures. It was like, as I said, it's like you found the 5th Beatle was living next door to you or something. I remember the hairs in the back of my neck were standing up here. And I will never be condescending to an elderly person again, I said to myself. But it gets, there is more to it than that. So here is the actual purines that she elucidated. This is adenine, and this is guanine in all its glory. But in addition to that, June had not only said that this is the dimensions for Watson and Crick, these are the pieces, the shape of the jigsaw puzzles that they were going to work with. She also said, I think that they're linked together in the crystal structure by entities known as hydrogen bonds. Broadly speaking, there's covalent bonds in chemistry, which are very tight, rigid, unbreakable bonds. And then there's the idea of hydrogen bonds, which is a weaker sort of bond that one can sort of disrupt quite easily. And so she felt, if you look at this model of her adenines, this is shared by Mike Glazer, who has it at Oxford. Am I allowed to say Oxford here? So it is Oxford crystallography collection. He was also at Cavendish. And this is a model based on June's work. And one can see vaguely, if you squint your eyes, you can see there's an adenine, but the critical piece are these hydrogen bonds here as well. And so she was saying, this is what they look like. And they're linked together in this fashion, she thinks, by these hydrogen bonds. And this matters because at this stage, so she decamped. She did this work by 51 and she moved on, as I'll tell you later. But Francis Crick and James Watson were working away at it in 52 and 53, and they were a bit stuck. They had seen Rosalind Franklin's picture 51, and they realized there was a helical structure. They're trying to figure out whether or not the bases pointed out or pointed in. We need some due diligence because I am a pediatrician in Ottawa telling the world that, hey, look at this. But here we have Durward Cruikshank, himself an eminent crystallographer, who I found also in my academic search. Other would call it Googling. But as he said, as Watson and Crick worked on it, one of the things they looked at was a possible base pairing bonding patterns that was derived from a wealth of crystallographic data. As he said, Watson was studying June Broomhead's papers and the thesis of 48 to 51 on crystal structure of adenine and guanine. Happily for him, the thesis was available at the Cavendish laboratory. And in the words of Watson himself, oops, sorry about this. I was drawing the fused rings of adenine on paper. He was staring at June's figures, and he was staring at actually this figure right here, figure 8 from the first paper. So here's the structure, and there are the hydrogen bonds. I was staring at that, and suddenly I realized the profound implications of a DNA structure in which the adenine formed hydrogen bonds, and similar to those found in the crystals of pure adenine. And then he went on to postulate that adenine would bind to adenine and guanine would bind to guanine, etc. And then he would have a double helix. So he thought it was something like this. There are the 4 units, and it would be sort of like with like. But he rapidly realized, because of June's work and the size, on the left-hand side, one can see the adenines and the guanines, the complicated ones that she elucidated, and then the more simple cytosines and thymines, which had been previously known. And these are bigger. There's just more molecules than these are. And so it didn't work. There were buckles and folds in the double helix. And so they were stuck. This is within the first 24 hours or so of him having this epiphany. But then talking to a guy named Jerry O'Donoghue and just thinking a bit more about it, this is Watson's true brilliance, I have to say. He saw that adenine linked to thymine and guanine linked to cytosine, once again using June's sort of steric coordinates, was the same size. And then in a sort of a complete epiphany, realized this fit with Erwin Chagraff's earlier observation, an American biochemist, who said, you know, when you look at the DNA from any creature, any source, the amount of adenine and thymine and the amount of guanine and cytosine, they can go up and down. But the exact amount of adenine always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. And so in retrospect, you say, oh, duh, they must be linked together in order to have them in lockstep as far as concentration. And that exactly was the case. And so that this led to the famous picture, which is actually taken from the paper with oddly helical misspelled, I think, there. But nonetheless, we see a thymine with adenine and cytosine with guanine, the so-called bases in the middle there. And here, this is the actual structure, and here are some of the, and on the inside, you can see the guanines and adenines that Watson and Crick used. And they were actually taking this welded structure and trying to putting it together a bit like this, seeing if it fit. That was the level of the analysis that they did. Critically, the fact that these hydrogen bonds existed here meant you can peel them apart, and put them back together. So for replication, you just take these apart, copy it, and you get the new copy of DNA, something which is used in polymerase chain reaction. When you do forensic DNA, the idea of binding one bit of DNA to another is exactly how it works, this so-called hydrogen bonding that June had pointed James Watson in the direction of. So yeah, anyways, it was just an unbelievable thing that I stumbled across. So half my time is up. I'll just tell you a bit about June, the June that I know. And Jane is going to kill me, but her daughter is listening in as well, so she can correct me if I've got any of it wrong. She was born in June 7th, 1922, which if you do the math, she would have been 100 this Thursday. She died this past November. Her mother was Florence, a devoted mother and a housewife. Her dad, Frederick, was an engineer in a coal mine. She went to a local girls' school and at some point determined she was going to be a scientist, but she was the only one at the senior level who wanted to be a scientist. Thus, she was alone in the upper class. And then in 1937, the New Zealand on the atomics, she read the death notice. She read his biography, June fashion. She read the biography, where he had worked, and determined then and there at the age of 15 she was going to go. And she had, although Latin wasn't taught at her school, she had to find a tutor. But June being found a way and accepted to Newnham in 41 on a full scholarship, and the first one ever to do so from her school to go to Cambridge. And she clearly was given a research studentship after she graduated in 44, but being the end of the war, she was dispatched to teach for 2 years in physics, but returned enthusiastically to Cavendish to do a PhD under Will Taylor. A brief word about Will Taylor. June was the sole author on her papers. Taylor supervised her along with Bragg. June was clearly autonomous, but at no time in 2022 would you ever have a graduate student that your name did not appear on. So that really, it was a gentler and I think nobler time in some ways in those days. The fact that June was allowed to be fittingly the sole author on her papers. Here she is, I think, son Robin tells me this is graduating as an undergraduate on her way to teach physics. She then returned to the Cavendish to pursue the PhD under Taylor, and she had not initially an undergraduate heard about X-ray crystallography. She knew nothing of it, but as she said later on, by pure serendipity, she studied the courses that you needed: physics, chemistry, mineralogy, and mathematics to do so. And then it fit perfectly with her personality. It required great patience, quite a lot of intuition, huge focus. And as she would say, one would collect one's experimental data, retire to the desk, and for many hours of thinking and calculating. And the June that I came to know was one who just loved to have complete autonomy and to be able to think carefully and cleanly about a subject. It's like some incredibly complicated game of Tetris, demanding sort of superhero numeric skills. I'm not very numerate myself. I've never understood it, but X-ray crystallography, but I think that's what it was. And here she is on the far left here as one of the only two that I can make out women at Cavendish. And there's Max Perutz and a couple other Nobel laureates buried in here as well. There are 17 Nobel Prizes that came out of crystallography in the Cavendish, so it really was Mecca. And here is a picture of a young Scotsman, June on the right, Bill Cochrane on the left, who was just a brilliant and a lovely man basically who I think was in love with June. He really, they had a very strong relationship. He helped her doing so-called Fourier transformation of her X-ray data. X-ray, she helped with X-ray. She also helped with Francis Crick as well in doing the alpha helices. He helped with that. Bill was involved with over 3 Nobel Prizes himself, helping people out. And anyways, a very important friendship for June while she was an undergraduate. That was while she was doing her graduate work. I have to say, Jane, full disclosure, that June did tell me that there was a possibility of matrimony with Bill, but he was such a mild and gentle personality that she was worried she would turn into a harridan and a virago if she married him. And so she chose the more clear-eyed, forceful thinking George Lindsey, who she loved dearly. She went on to work with Dorothy Hodgkins, another Nobel laureate in Oxford, working on the incredibly complicated structure of vitamin B12. This is one of the three papers that came from that. And then she moved into Ottawa, where she did work at the National Research Council and elucidated the structure of codeine. She shared the data that she had at that stage with a graduate student of Dorothy Hodgkins, so she could have a parallel publication because they were coincidentally working on the same project, just showing sort of both how she and Bill Cockman, they had no discernible egos when it came to their science. Another paper, another vitamin B12 paper. At that stage, George had moved to Montreal, and there was no more X-ray crystallography at that stage, and so it just she had to step away from it, which I think was a bit of a blow for her. Just how highly treasured she was in this area you could see with this letter from Bill Bragg, where he wrote a rather poignant one saying that Max Perutz had been struggling with his structure of hemoglobin and I believe insulin at that time. And as he said, we badly need your hands to tackle this naughty crystallography problems. It would be right up your line. He wished very much that she would come back, but her major focus was, and Jane, you're going to cringe, were both Jane C. Lindsey, who is now a very successful statistician at Harvard, working on HIV projects, as well as Robin Lindsey, who is a professor of economics out at UBC. She took time off to actually, there was a student strike, a teacher strike in Ottawa, and she actually came out of retirement to teach. And coincidentally, she taught my brother-in-law, Philip Hampson, who went on to go to MIT and Imperial College. And he said she was the best teacher he had ever had, bar none. So she really was remarkable in that facet as well. And then, and life went on, and Jane can talk about how she felt. She is an absolute role model for STEM and this idea of science. Philosophically, she was determinedly not a feminist and felt that her proper place had been at home, and that basically men, she felt this made the young women around her cringe somewhat. Men were really in some ways better at solving scientific problems. And as Jane has said in the past, she's not sure she would still, if she was born today, she would still adhere to those views. Life for her changed dramatically when I stumbled across her, because I really was taken by the story and at some extent offended by the fact that the world did not know. So I wrote a piece about it. She was on the CBC, there are things in the newspaper, and there was a huge amount of attention brought to bear upon her, or at least a substantial amount of attention. And in the end, it looked like it discomfited her. But I didn't care. It was just such an important story that I just thought it was one that one needed to tell. I did care, and I felt badly, because crazy people were writing her letters from the States asking for pictures, etc. And there were going to be films put into films, etc. And she seemed it seemed to make her uncomfortable. But as Jane can attest, the day before she died, she asked me, she bid me to her bedside, and then she said she thanked me, which was hugely important. That basically, in the end, at some level, she was not totally displeased with the fact that there was some recognition of her marvelous work. Yes, I was happy for whatever small part I could play, but we are not finished yet. And Jane and Robin have started a fund for this remarkable woman. And also our hidden agenda or not so hidden agenda. This is a photograph of a caffeine molecule that's in New Jersey at some, I think Rutgers on the quad. And Jane and Robin and I hope all of you would think that it would be great to have a picture of either guanine or adenine of June's guanine or adenine. If you have a building for Ross's important work, Ross Franklin, it would be lovely to have a sculpture of this nature in Newnham to remember June and her work. And there we are, Margaret. I finished 5 minutes early. And I perhaps raced through it a bit too fast, but happy to take any questions that might be emerging. And I can't see it. So maybe it's just Margaret, it's just you and I and Jane and my wife, Beatrice, who's on now, talking to each other. I'm not sure even if there's any audience, but anyway, happy to take any questions.
I can assure you we have a large and very interested audience here. And
tell me they're standing, but you can tell me they're standing up and applauding now, and I'll believe you.
Your insights are fascinating, and all I can say as a non-scientist, thank you for such a clear description of June's work, and I'm sure other non-scientists in the audience would appreciate that also. So now I think we have time for questions, and if anyone wants to raise their hand, we'll bring the microphone over to record the questions. Well, Alex, I have a question for you, and it is, was there any communication between June and Crick and Watson during that time when they were studying her work and creating their ideas?
No, there wasn't. They sort of, Bill Cochrane stayed on in Cambridge and as I said, was a central part of helping Francis Crick figure out what alpha helices, how to analyze them. And so insofar as both June and George remained friends with Bill, there were some peripheral interactions, but no, there at no point had they reached out then or after. But I think it's true, whether it's Ross Franklin or June or some others, that Watson and Crick were just very, very focused on the scientific project, taking the data at hand. And if you weren't Linus Pauling or Morris Wilkins or one or two others, they didn't really talk with others. They just took the data. And I have to say, in a very admirable way, they put together the jigsaw puzzle in a really inspired way. But no, there wasn't a lot of talk. And then for June, it was almost determinedly that she loved her time at Newman. She was never so happy as when she was doing the crystallography, I think. But she had moved on to a new chapter of being a mother and a wife of George, and didn't that scientific part of her, of which she was clearly so obviously proud, as one could tell at the very end, what she said to me, was, if not suppressed, it wasn't acknowledged. And so, no, there wasn't a lot of communication. And in point of fact, when those molecules, they knew it was the hereditary material, and so that made it more interesting. But when Taylor and Bragg assigned it in the mid-40s to June, it was just another complex and one of the more complex molecules that they had a bit of a, I don't want to say assembly line, but they had, you know, a bit of throughput as far as molecules they're studying, and these were just two more. The fact that they were, in my view, two of the most important molecules in the universe, or at least our universe, was neither here nor there. And if you look at the discussion in the papers itself, filled with sort of mathematical aperçu and talking about this and that technical, no acknowledgement that, you know, its role in DNA itself. So they were pure scientists, really pure scientists in that regard.
Was there any particular reason why she moved over to Oxford? Was it just that she was interested in broadening the scope of her work?
Yes, well, she was bid to. I mean, in those days, she was still a very young woman. And it was felt by Bragg, I think, that, in order to further her maturation, Hodgkins was already a force in the landscape over at Oxford, and they thought it would be best for her further development to go over and work with Hodgkins in Oxford. Ironically, she had an enormous respect for Hodgkin and fondness, but Hodgkin was so good at what she did, was a real genius, that that part where June could sit and quietly look and put the pieces of the puzzle together were taken away from her, in a sense that Hodgkin would come in with huge enthusiasm, stare at her data, and then click, 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 put it all together in a way that I think, that June found a bit vexing. And so it was that at that stage of her life, although as she kept in touch with Hodgkins, and they published into the 60s on that very thorny problem of vitamin B12. At that stage, she decided to marry George, to go over and move to Ottawa, and move on with her life at that period.
Just following on from Margaret's question, did June know Rosalind Franklin personally?
She did not. She did not, and no, Roz, and yes, she did. I have to choose my words carefully. She did not know Roz Franklin. She felt, how do I put this? She felt if you looked at unsung heroes in the DNA story, her friend Bill Cochran was perhaps the one that was the most seminal, and so. But she never met Franklin that I'm aware of. Jane, you might correct me, but certainly she obviously was aware of Roz. But it's interesting, the whole dynamic. June came quietly, did her work, left it there, and then moved on, Oxford and then Ottawa, and so really didn't interact, and it didn't speak of it. I mean, the thing is, me having to go on her 95th birthday, when she was 95, and her saying it, I would be telling everybody all time, every day about this stuff. So that was the thing that just blew my mind. Anyway.
We have 2 more questions coming.
Okay.
Alex, thank you very much for an excellent talk. Really appreciated it. As a Yorkshire-born person myself, I noted that you said that she was from Yorkshire.
As were the Braggs.
Right, okay. I didn't know that either. Do you know where in Yorkshire she was from? Or possibly
Okay.
Jane can tell us.
Jane will know. Jane, you can tell us where in Yorkshire you can come. Jane said you didn't work. Yes.
It was Doncaster.
Right, thank you.
Great. And where were you from, Yorkshire?
I was born in Skipton, but my family are from the Dales, and confessions time now. I actually was brought up mainly in Lancashire, but don't tell anyone.
There we go. We won't tell us all. Great.
Were her papers cited by Crick and Watson?
Eventually, I'm not in the original one, but they were in the 53. There was the first 2 papers. They held one so that Roz could and Wilkins could get one in Nature, but then there were a series of after the first one, and her work was cited at that stage. And so, but they're not citation classics when you go back and look at how fundamental this is and how inflated things are. Some are cited 168 times or something. So to this day, I'm underappreciated in my view.
We have another question at the front. Right here.
From the later discovery by Crick and Watson, what implications did her discovery have in the real world?
So sorry, Margaret, I didn't totally get what implications.
What implications did her discovery have otherwise in the world?
And so, okay, I mean, so her work, as the Latins say, was a sine qua non, without which would be impossible for Watson and Crick's double helix. So basically, that was clearly, the most important thing. her other work, she worked on the actual the bind, and this is in her PhD thesis. So not only did she look at guanine and adenine, but she also, in the earlier chapter, looked at how water is complex with what they call cations, positive ions. And then she helped with the incredibly complicated structure of vitamin B12. And then she helped elucidate the structure of codeine. So for somebody whose scientific career was essentially, I think, 9 years or so, it was a pretty impressive epistemologic output, I would have to say. And then I would hope her example, along with Ros Franklin's, will help inspire young women to do more science, at the risk of sounding trite.
Jane, we've all appreciated so much the research and work that Alex has done to let us learn about your mother and her wonderful achievements. Is there anything you would like to share with us?
Well, I'm extremely grateful to Alex for the serendipitous meeting that he had with my mother when she was 95, because, you know, as many children, I think they don't appreciate their parents and what they've done, and they only look at their parents, you know, from making dinner, and they don't realize their parents had a life before. So I know Robin and I are extremely grateful to Alex for giving my mother this sort of late fame in her life. And she did appreciate it. I think she was a little puzzled by it, but she really did appreciate it. So I think it's pretty cool and exciting that she had that at the tail end of her life.
And how about my dime store psychologizing about her, etc., and her view of women in science? Was that your view as well, Jane?
I think, yes, I agree. I think she was really torn by it. And, you know, it's an unknowable question. Yeah. She would have done the same thing if she'd been born 50 years later. I don't know. I don't know that she knows.
Yeah, yeah. Well, very few children sort of are aware that their parents have done seminal work in the discovery of the most important molecule of life, because most parents haven't. But you had a remarkable mom. Yep. So yeah, I mean, if you can't achieve scientific greatness on your own, have the grace to recognize it when you stumble across it, I guess it might. Anyway, Margaret, thank you very much for letting me.
I think all of us here in the room, and indeed all of us here in Newnham, really truly applaud, and I mean it, applaud your effort and all the research you've done to let us get to know June and all her work. So okay,
well, quid pro quo, everybody. Oh, thank you very much. But quid pro quo, everybody get out and push for that sculpture in the quad. We can all have co-ownership of it.
Yes, indeed.
Okay, all right. All right, thank you so much.
Thank you.
Okay, bye everyone.
Goodbye.
Thanks.